Yes, thank you very much. It's a great pleasure for me to welcome you all virtually. I can see your faces, but it's great to see so many of you, people interested in the topics that we present at our ICM virtual seminars in computer and computational science. First of all, I would like to acknowledge our supporters, partners, and a patron. Our honorary patron is National Information Processing Institute, or OPI. Media partners are Datanami, HBC Wire, ITWIS, Sztuczna Inteligencja, Enterprise AI, and Computer World. I thank you all and acknowledge their support. That is one channel of reaching out to a wider audience and secure interest of people. And as you might realize, the competition for your attention is extremely tough, intense now in the times of pandemic, and there are lots of very interesting events, seminars, talks going on. We are changing the mode. We promote science and inform people about the new results. And let me just briefly say a few words about ICM seminars. The origin of seminars stem from our conference, which we have already run 6 times, 3 times in Singapore, 3 times in Warsaw, last time in March this year. And the last edition was fully virtual. We had about 50 presenters over a very intense full 3 days of conference. I encourage you all to check our website, the conference website, because we are very fortunate to each time attract outstanding speakers. And that's of course also a case with the series of seminars that came out and in a natural way after the conference, since we already had the interested audience for the conference. It was over 1,000 people from 67 countries. So we thought, why not continuing this and to bring the most interesting speakers, thinkers, visionaries who basically create a new vision of the world in the computer space, in the space of computation and computer technology, computational sciences. So here's the list of 11 speakers who gave talks, and all those lectures, these seminars are recorded, and you, of course, you are most welcome to pursue the topics and see how fascinating the topics that we had previously were. But today we have something really special, and I'm very excited, very happy. Our today's speaker is Dr. Hiroaki Kitano-san, who is the president of the Systems Biology Institute and president and CEO of Sony Computer Science Laboratories, and also a principal investigator at Open Biology Unit at the Okinawa Institute of Science and Technology. Dr. Kitano is known for developing AIBO, artificial intelligence robot, a series of robotic design, dogs designed and manufactured by Sony, and the Robotic World Cup tournament known as RoboCup. I had the great pleasure of meeting Dr. Kitano-san many years ago in Singapore, where we had short interaction, but ever since, and even before that, I was really intrigued and fascinated by the technology and the concept behind this technology and the idea developed by Systems Biology Institute. And I was particularly interested in one of the products being developed by SBI, which is called Garuda. And I hope we'll hear something about Garuda, because I think it's a high watermark. It's a gold standard of how certain solutions should be developed and presented to users, how extremely useful they are. So we'll hear that. But of course, Garuda is not the only product of, and not only the single topic of development at the Systems Biology Institute. And I also have a great pleasure to announce that not long ago, about 2 months ago, ICM has signed MOU with the Systems Biology Institute. We've been involved in several discussions over the last couple of months, and I can see that there's a very interesting, great scope of interaction. What's very important for us at ICM is that we are not only recipients of this great technology, but hopefully we may add a little bit of our own, discoveries and ideas that have been developed over the years at ICM. So I'm looking forward to very fruitful collaboration between Systems Biology Institute and ICM. I think it will be exciting for all of us. And with that, it's a great pleasure to invite Dr. Hiroaki Kitano-san to give a lecture titled Nobel Turing Challenge, Creating the Engine of Scientific Discovery. Thank you.
Okay, thank you very much. Well, thank you very much for a very kind introduction. This talk, I would like to share with you idea and then a vision for the revolution in the biomedical science and then artificial intelligence. And then I will talk about like a new challenge, what we call the Nobel Turing Challenge, is to create like an AI system to autonomously discover very significant scientific, make a scientific discovery. And we set a specific goal of creating an AI system by 2050 to make a major scientific discovery that worth Nobel Prize in physiology and medicine. And I think, I wrote a paper published in AI magazine back in 2016, and then I got a couple of Japanese papers as well. And but when I say it's like AI will make a discovery and probably win a Nobel Prize, then when I start talking about this, one of the people in the front row in the audience at one of the talk come to me and says like, well, I mean, that sounds very intriguing, but like if you look at the Nobel's will, and they will give it to the person, and they will not going to give it to the machine. Yeah, okay, I understand that. But I think I'd be then I come up with an interesting tweak on the challenge, and which is the Nobel Turing Challenge. Is the challenge is like not only the machine make major discovery, but at the same time, would machine would be behave like a life, like a human life. So like a Nobel committee won't be able to ever tell that discovery was made by the human or not the AI system. So I think it's a very interesting challenge, which is actually a major scientific discovery challenge at the level of Nobel Prize. I mean, Nobel Prize is, just like I use Nobel Prize as a symbolic example of what is the level of discovery we are aiming at. At the same time, it's a Turing challenge. I mean, a Turing imitation challenge at the highest level of the scientific activities. And then actually, the Edo Feigenbaum and Stanford professor has like a Feigenbaum challenge, which is a variation of Turing challenge. It is to actually make the machine at the best human expert level, and which is going to be indistinguishable. So this is actually variation of Turing challenge, Feigenbaum challenge at the scientific area. Okay, so but still you might ask a question that like, well, I mean, you probably able to tell what is the AI and what is not AI, and then probably people just wants to distinguish that, and then people just use the result for the human scientists. But if you are knowledgeable about like what's going on in Bitcoin and the blockchain, a whole this started with the one paper in a blog post posted by the person named Satoshi Nakamoto. If the Bitcoin blockchain changed the economy, and the Nobel Prize in economics have to give an award to the Bitcoin blockchain, the committee have to give the award to Satoshi Nakamoto, because this is the beginning of everything. It is very clear that this paper posted on the blog is the beginning of everything in the Bitcoin blockchain. But the problem is no one have ever met Satoshi Nakamoto, and it's like a very clearly Japanese name, but we don't know if this person is actually Japanese or a group of individual or like whoever it is. And it's this the identity of the author is absolutely unknown. The only reason we believe this is not created by the artificial intelligence. We believe AI is not good enough at this moment. So I assume that a major biological discovery has been made and a paper posted in the blog. And if you do go after the paper, do the experiment, it beautifully produced, and then it will be the game changer in the biomedical science, for example, or like a physics or any other field. I think that people start using it. People actually develop the new theories, the new science, new industry out of that kind of discovery, even if the author identity is not known. It could be the AI or it could be the human, but probably it doesn't matter because that's how things happen in the Bitcoin and blockchain. So I think it would be a very interesting challenge to create a machine to make a major scientific discovery and able to write a paper, communicate with the people, and probably eventually win a Nobel Prize. Of course, like by that time, machine will make a major discovery autonomously, and probably like every 3 days, like you got a big discovery. So like a value of Nobel Prize will be in question at that time. But like I think this is very interesting, a challenge. Okay, so in order to do this, we're going to have two sub goals. Okay, obviously, first goal is to have a machine and the robotic system, because we probably have to do the experiment as well, to able to perform very good science and make major discovery. Okay, and this has been done autonomously. It's autonomous system rather than a tool, AI tool, to make a scientific discovery. Okay, that's one. So of course you have to make a big scientific discovery. And the second, machine must be able to make a strategic choice over the topic of the research and probably communicating from publication, obviously, or probably like a presentation at the conference, maybe as well, and then able to tell the reasoning behind the discovery, or able to value what is important. And this is very interesting in a way that the we can make a lot of discovery, but what makes some discovery more important over others is discovery if that makes like an impact to the human civilizations. Then we consider that's very important. Like building, exploring new opportunity or cure the disease, and that's all aligned to the human civilization, because like human being is the one who evaluate the value of the science, because that's more very anthropocentric value system, but that's the reality of our society. So I mean, curing, for example, C. elegans. For example, if you find some disease in C. elegans and you find a cure, okay, that's important. But at the same time, we value the discovery if that translates into the human disease and its cure, right? So I think it's more the human-centric. So that means the machine have to value the discovery consistent with the value system of the human society. So like a machine need to learn and able to align the value system of our society and able to communicate why that it's important, and probably invest more computing resource, invest more experimental resource along that line. So like that means not only machine make a major discovery, machine will be able to judge which one will be major in the context of human civilization. So that this is the two big challenges. At the same time, I'd like to caution you that I'm not really claiming that every discovery will be made by the AI system. What I'm arguing is AI system will make a lot of discovery, and some of them will be very important in the context of our civilization. That's what I'm claiming. So like I think it's still the human will make a major discovery as well, but the AI system will make a major discovery comparable or even more significant than the human discovery. Okay, so that's the point of the challenge. So let me why I come up with this idea is, let me go back to the one of the very first biological research I had. That's I go back to the early 90s. I start getting involved in the biology after spending years in Carnegie Mellon and working on the AI and the parallel computing. And then the first research engaged in the aging of the solar systems. And then Professor Shinichiro Imai, who now the professor was in St. Louis at that time, it's in Keio University in Tokyo. And then we discussed, and then he actually said like there are quite a number of the data, but at the same time, what the people talking about the theory behind it doesn't seem to be very consistent. So maybe we should work together to come up with a computational model to be able to identify what is the real mechanism behind the cellulousness and cellular aging. Okay, so we have like some data. At the same time, this is the pre-omics day, so like the data was rather sketchy and sporadic. But we come up with like some model to be able to so like then in the process, what we actually found is the basically we come with the hypothesis, but like most of the biological research is trying to identify the mechanisms behind the biological process. Either it's a disease, either it's a fundamental process like a cell cycle, or cell proliferations. Then what you need to associate is like with that process, what is the things behind it? Okay, that means what is the molecular mechanism? What is the interaction in between them? Interaction in between the cell or organs, and then what molecules or genes are getting involved. So those are the things we want to discover. That can be like deeply stacked or like a very single mechanism. But that's essentially I wouldn't say it's everything, but the major part of the biomedical discovery is to identify that relationship. And then we come up with like a number of the possible molecular mechanism behind the senescence, and then we come up with like a relatively simple computation model and to the standard, and parameterize that, and then what happens if this molecular mechanism with a range of parameters, we come up with like a 500,000 different cases, so like a model configuration and the parameter scans, and then run the simulation for like 6 months, 7 months, at that time, and then which model will actually fit the experimental data. So this is very much computational biology, system biology in the early 90s. At that time, like there's no system biology field, because I come up with the name back in 96 or 97. So like this is before the system biology start talking about system biology. So but at the same time, with the 500,000 simulation runs, we come up with like a very specific mathematical structure which can consistently reproduce the various data available at that time. And if you are talking about relatively simple mechanical modeling, and people might say like, well, I mean, if you tune the parameter, there will be many different models which can equally well fit to the experimental data. But things we are lucky about is there's only one mathematical structure which can explain all the data available at the time, and which was like there's two honestly the counter, but like a master control genes, and then two process, and then two independent effector gene group, and we call the one process S process, another the C process, and then effector genes, outcome of the genes like S process gene will impact in a paracrine manner, and then go to cell arrest. And the C process genes is function as a cell autonomously, and then arrest of the cell in the body of the cell that express that gene group. Okay, and that's a mathematical structure, and then we have to interpret that into the biology, and then we come up with the heterochromatin structure, and then probably paracrine, secretion factor impacting the neighboring cell. This time, in early in 90s, cell senescence was believed to be very cell autonomous process. So like, when I talk about paracrine, people say, a lot of people was very skeptical on this. But like this was actually the only mechanism, and it's biological interpretation which can reproduce the data. Okay, and so they come to like, and then we submitted the paper to the science, I guess, but like they come up with says like, can you come back with the gene? So like we decided not only the mechanical structure and then a mathematical model and the possible interpretation of molecular structure, molecular mechanism, let's identify the genes. And then Shinichiro, we had a pretty good estimate on the idea of what is the most likely genes, and which is ESA2. Because So 2 is the heterochromatin remodeling, factor, and then also there's a homologue in the various different species. Since it's so fundamental, we consider this has to be involved in heterochromatin, and it has to be preserved among the species. So we mentioned this about this, possible hypothesis, and then published a couple of papers, and then Dr. Imai moved to the MIT professor in Garantel's lab and experimentally, verified actually the SIR2 is a not dependent histone deacid and controlling the longevity of the yeast. And then human homologue is what we call the sardines. And so this is the one story behind the sardine family. Okay, so this has been really good. And then after that, Professor Imai, continued the mouse experiments and identified overexpression sardines, and then intake of the nicotinamide molecular acid, which actually impacted sardine expression level, were able to expand lifespan of the mice. And then female and male, and the female is about 20%, male is about 12% lifespan expansion. Okay, and then we'll go into the clinical trial in human at this moment in Japan, and so it's getting to the really successful project, I guess. But since we started back in this project back in '94. Took us over 20 years to get to the human clinical trial. And then all the process, hypothesis generation and computational model, and interpreting biologically, and then the verification using a mouse, and then all that, and then go to clinical trial. Only place we did actually very systematic, and we saw nearly almost exhaust such is the computational model I mentioned. And the hypothesis generation, we thought about this molecular mechanism is possible, this is possible, but I think we're probably not exhausting all the possibilities. And then after that, we have to do the biological interpretation of mathematical model. And we made the best, in reasoning out of that, but we're not sure. We actually try to cover all possible interpretations, but the most reasonable. And so we rely on our knowledge and our scientific intuitions. So in the whole process, only place is in the computational modeling, and all other thing, we have to say that we has been very lucky, and that's why we are getting there. But this looks like a very fragile process, and that is actually what's going on in science. And that's why I consider the scientific discovery is still the pre-industrial revolution level. We now have the high throughput genome sequencer and omics system, and probably live cell imaging, a very sophisticated measuring system, and probably a large scale data analytics tool. But at the end of the day, if you remember, think about your own research. At the end of that, you scratch your head and try to figure out, okay, what this data means and what is a possible hypothesis, and you have to think about it. Okay, because this process, I think it's a joy of the science in there, but still, this process is not changing in centuries, and this process need to be automated. And need to be, have to have a computational basis and how we can make scientific discovery. So therefore, we can understand what you mean by the serendipity, what is actually the implication of the discovery by accident, or what, we talk about scientific intuition, what that really means. If you look at the interview, for example, Nobel laureate, surprisingly, a very large proportion of the winner says, "I was just lucky." We screw up experiment, because of that we find something interesting, or things like this. So what does it mean? And then people talking the serendipities. Okay, and so after I proposed this system biology late in 90s, did quite a lot of research going on. And at the same time, if you look at the reality system biology, very precise mechanical model is relatively small to medium-sized model. If you go to large-scale model, what happens is that it's very close to, or probably almost identical to bioinformatics, or the statistical analysis or omics, rather than go into a mechanical or very detailed model at a large scale. And that's the reason for that, because that's actually coming to the conclusion, hit the very limit of our cognitive process and social behavior. So a system biology, in a real sense, a large scale system biology, it's probably not suitable for the human being alone to do that. So it's probably the science for AI, or AI human hybrid system is more suitable to pursue the systems biology. Okay. And the reason for this is because our cognitive structure, we think and reason and communicate in a natural language. And our cognitive structure enables us to reason and hypothesize, but that process actually is limits our cognitive capabilities. And then, of course, nature of the scientific community and amount of data actually also limits our capabilities. Like a few limitations, issues there, but like I can't really go through the everything. It's taken so long. But I'll show you some very stereotypical example. Like this is very famous. But like this is actually a paper published like a few years back. But like in the biomedical field, 1.5 million paper published per year, and this is almost 10 years ago. Okay, so I think today we have probably over 2 million paper published per year. That means like about 5,000 paper per day. And then if you are in a field like oncology or immunology or neuroscience, I think hundreds of paper by day, and there's no way you can read them all. Okay, so you believe that nature science and PNS and so but like you never know where the important paper lies. Right? So like we pretend that we know things, but like we're actually accessing very fragment of what we have discovered, and then what we discovered every day, what published every day, and then keep going there. Okay, and then for example, we want to try to assemble the knowledge discovered. This is like a cell signaling map that we created like almost 10 years ago. This is the most comprehensive signaling map of the eukaryotic cell, and we actually map out every signal transactions and very much in detail. And read into the material methods and all that took us like years to build this. And then we have like this one is E-cell cycle. This is very much the most detailed map of the E-cell cycle. And this is 2-RAC receptor. And almost over 10 years ago, and it's very difficult to update. That's part of the problem. And this is about the Alzheimer-related pathway. Okay, so the reality is now we're improving this back. At that time, we built this, and I was really labor intensive. Like we read the paper very much in detail, and with a lot of teamwork in the curation, and then we get the post-its and all that, and then we built a mental model. And so like the problem with that, you can build one, but you can't keep updating it at a high quality, because like okay, we can't really keep doing this. Psychologically, it's very difficult, and then sociologically, how are you going to guarantee the career path of the person who keep reading and then building a map? And so like all kind of issues there. Okay, another issue I would like to mention is what we call the minority report problem. Is if 99% of report indicating A execute B, for a very simple example, and if we have 1% report indicating the other way around. Okay, so first of all, if this is 100 paper, maybe we notice that. But if it's like 1,000 paper, only few actually saying the something different, and it's very questionable if we ever notice that, or if we notice that, we'll probably just dismiss that. Well, I mean, this is probably some experimental error or unreliable report. But like it's possible that reporting something very specific and a specific condition. Something unexpected happening. So how can you tell that? How can systematically do that? You can, do it for one example. Like, can we do that in scale? And that's the whole game here. Like, can you do that in scale? Okay, and then also, human language use is highly contextual. That means that we always suffer from the cognitive bias, and particularly, for example, like a clinical, setup with the clinical reasoning, and there's a lot of bias, which is a very serious study, field of study. And Alfred Kojivsky, the founder of the field called general semantics, says map is not territory. This map is actually human representation. Territory is a reality. So, like, if you describe the reality in a human language, it's no longer the identical, and you communicate, and then the recipient of that information will map out, create the mental model, which is also different. So like also this this alignment continues, and then we actually use that for the scientific research as well. And there's many examples this alignment hampers all the proper scientific reasoning as well. So like we have like few more examples as well, but like I probably stop here. And there's a limit of human cognition. So what we're trying to do is use the power of computing, power by AI, to overcome or complement the human limitation of human cognitions. So the so what we're trying to do is actually creating engine for scientific discovery. And of course, scientific discovery is not new in the field of artificial intelligence. And soon after the field was created, the group of researchers at the Stanford University, Joshua Rederberg and Bruce Buchanan and Fagenbaum, come up with the AI system called Metadendral, and which using mass spec data, and it can infer what is actually the chemical which is present and create a hypothesis for that. And under the many scientific discovery program there. And recently, Professor Ross King and the University of Manchester, which is Chalmers in Sweden right now, come up with the Adam and Eve and system, which create a relatively simple East genetics hypothesis autonomously, and then design experiment, execute the protocol, and actually do the experiment using this robotic system and verify the hypothesis. This is one of the first closing the loop system, which a system autonomously generate the hypothesis, do the experiments, and verify that fully autonomously. Okay. So, I mean, this is relatively limited in terms of the hypothesis generated, and then also the kind of experiment which can exercise very much specific. But still, it's getting there. Okay. And so, in order to pursue this challenge, my working hypothesis is that scientific discovery is a massive search and verification in the hypothesis space. Okay, so then the question is how efficiently we can execute it, and how extensively we can actually create the hypothesis space to fill the possible space, and how effectively and how certainly we can verify that. So that is the kind of thing. And then you might actually ask the question, is it actually true? I mean, it's scientific discovery, research, and verification. I mean, theoretically, that should be that. In reality, what's going on could be something different. But so I look at the number of cases in a novel quality scientific discovery, and then one example on the left side is the example of the Professor Shinya Yamanaka in Kyoto University on the discovery of iPS. Cellular reprogramming to pluripotent state, and then he initially searched for genes that may actually able to program the terminal differentiated cell, and then identified 24 genes from the phantom data, which is a big Japanese project. Identified 24 genes which could potentially be the reprogramming factor. Okay, and he actually interestingly put them all, and he found out that he reprogrammed the cell, and then go to the live analytic experiments and identify the minimum viable set. It's like a 4-factor of be able to reprogram the cell, and that was called Yamanaka factor. But basically, this is search and optimizations. Okay, another example is Professor Shirakawa's conducting polymer. That's in chemistry. Is that he received an intern student from the Korea, and he wanted to do the experiments on the polymerization process. And the student actually, this intern mistakenly increased the put the chemical at extremely high concentration, which is not supposed to do. Okay, 100 times or 1,000 times more dense one. And then the result was interesting because it's created a thin polymer. Okay, thin film. And then Professor Shirakawa found out, whoops, what's going on here? And then they started optimization to what is the optimal condition to form the high quality thin film. And then with the Professor Adam McDermott and he got them, and then convert this into the a very stable conducting polymer. And again, searching parameter and optimizing it. So this is a 2 example out of many, but search and optimizations and single step and multiple step has been the key. Okay, so it's kind of interesting challenge would be, discovery CRISPR-Cas, is a very distant thing if we combine together. So the archaeal cell immune system and then marine biology, and it's very distant. And it turned out that that combination give rise to the very interesting insight. Get to the CRISPR-Cas9. Okay, so I'm kind of trying to analyze what the process behind it. We may be able to formulate that, but even if it doesn't, it's a major, quite substantial part of the big discovery is the search and optimization. If you look at the whole process, so I think we should really start going from here, and then we need a technology platform to be able to do that. So in order to do that, we have to have like a basic functional module which is connected, because this is not like a without human intervention system. AI system will able to do the experiment and do the data analysis, right? So everything have to be connected, everything have to be online. Okay, and then we're gonna have like a basic AI module in each, and then we might have an integrated AI module to the experiments in a large scale, and then probably like an autonomous AI system to be able to, which will ultimately decide what to look into. Okay, and then our strategy here is why don't you do the connected research lab, which is very useful anyway, and then put in AI assistance for each process, and then integrate them and get to the highly autonomous system to be able to control all the asset, and they're able to identify what type, what question to be asked. Okay, so that's right. So then we started building the platform, and then as the Mike mentioned, we'll build the platform called the Garuda platform, which is a connectivity platform connecting all the data analysis tools and then simulator system, and then also equipment and all the devices. Okay, so that's our platform. And also we have AI stack called the Gandara AI framework to be able to use the AI module for each process. And that's something we built. In our Garuda platform, not only connecting all the data analytics pipeline and the simulation system, also we connect it with the wearable system, and then also with the experimental apparatus as well. And then this is one of the example using the Garuda. This is one of the Japanese national projects talking about the drug-induced arrhythmia. So in order to do that, you need integration or the known database, and also add a PK model, which is on the physiological modeling on top. And then now we have to estimate what is the dosage of the drug and all the physiological condition of the cardiomyocyte. And then we have the cellular level modeling. And then there's some mutation for the specific patient, like a Brugada syndrome, on the ion channel modeling, based on the genomic data. And that will be parameterized and then integrate into that model to get all the spike pattern, and then integrate over the whole organ model to be able to tell what is electrophysiology in a spiral with the regular propagation. And this requires number of software to be integrated together, and then we all on the Garuda. So that's things we did. And then also we are connecting with the experimental devices. This is together with the Shimazu Corporation on the very sensitive high performance mass spec. And then we provide the back-end data analytics and the pathway mapping tools as well. So streamline with the devices, and we're talking about using multiple devices to combine together to get the omics center, and it's fully powered by Garuda with the Shimazu machine. Probably can be the other company's machine as well, and that's what we're working on. And so we can have at this level. What we what we have is that the building is a basic functional module connecting together, right? And then next stage is for each process come with the AI. Okay, so let me use the example of the project we're working over last 10 years on the target drug discovery for the avian flu. Okay, so this is a work with Professor Kawaoka at Tokyo University and Wisconsin, and then identify is a drug discovery for the avian flu. Okay, so we have all the system biology oriented process, and then basically we go after the standard flow of the drug discovery is a disease-related molecular interaction, and then I use omics data, and I get the target candidate and prioritization and lead component identification and clinical study, and that very much standard process. And then for the disease-related molecular interaction, we want to understand the disease virus replication cycle. And then so this is the one we created for that paper, and this one is actually hand curated. Okay, so the progress afterward is that we're using a natural language processing system, obviously knowledge extraction system, to read the papers, and either deep parsing or like a BART or GPT-3, if we do that today, and then I'll be able to extract the knowledge from the very large volume of paper. And then one of the studies and the software we built a few years ago is to read the whole papers and extract the molecular interactions. Okay, so this one is the two-rack receptor map. Okay, and then background, which is white, is this is the gold standard, the human curated one taken years. Okay, and then green was among them how many of the interactions that the machine could have automatically filled in, and then we got the 95 to 97% coverage ratio over human curated one. So like we have a 2-3% missing, but like we are pretty good. At the same time, we notice the machine over-generating. Okay, the molecular interaction which is not in the gold standard model, and the machine is generating quite a number of interactions, and we thought that it's like an error. Obviously, because it's not in a gold standard. Then we start noticing that some of those interactions has been discovered in later days. Okay, whoops. So like a machine may be able to tell, identify the relationship which is not, recognized by the human curator, or like has not been well identified. Okay, so that's kind of interesting balance. So we keep going with the quite number of applications on the algorithm for the biomarker discovery using that kind of functionality, and doing the Alzheimer as well. And then see like all kind of images like what we do. So like we basically use the text-to-speech system, and we are now trying to integrate all the images in the paper to be able to assist the interpretation of text as well. And then we get all this galaxy view of the lot of concept, either the chemical or the process, or even the authors, who the researchers, and then all the relationship map. This is what we call the galaxy, and then each relationship has a meaning. Okay, so the recent paper along this line is very interesting. We published a paper, IGB transaction knowledge and data engineering. It's given the number of discovery, for example, like in this case, RDF4 is renal disease and fish oil, which is basically a cure for renal noise. Okay, then this was discovered back in 1985. Okay, so using the paper prior to that date, can we predict that relationship can be predictable from the paper prior to the year of the discovery? So like in case of RDF4, there was discovered in 1985. This is the first report came in 1985. So if we use paper up until 1984, can we predict that relationship? Okay, so with this case, like a 61% confidence, we can predict it. But like y-5, so like using up until 1990 and 1980, and very low probabilities, like it's almost impossible to put, like 4% confidence, very low. And it's interesting was like Alzheimer, like endometriosis relationship was discovered in 1989, and using the paper from up until 1988, it's 98%, a very high probability. But interestingly, with this relationship, even we use like a paper up until like a 1979, which is like y-10, 74% probability that we can predict that relationship. So this is interesting. So like this is a relationship, but of course, if we get the relationship, we can actually search around the possible molecular mechanism. So like this is interesting. We're getting to the stage that we can create a hypothesis, and then probably going to design the verification experiment. And so this is something very interesting. So like that something we did in this avian flu study, we did a hand curation, but we can actually do very automatic at this moment. No, perfect, but like a highly automated process, even able to tell us what are the possible relations which is not yet discovered. Okay, now we had to go to the omics study as well, and but of course, like we got the the structure. Okay, with the structure, and then we got all the molecular interactions, and then with the molecular interaction, we use the control system, control theory, to be able to tell which node will be able to control the process, which node won't. Okay, so we got the good mathematical formula like a hopcroft and then other ones to identify what is the key node which we should target for. Okay, so then we identify like this is sort of infection path, like all that kind of like what is the effect of the targeting one node, and then where the effect lies, and then that doesn't mean anything. Okay, so that means with the structure of the network, we have the theoretical list of the possible candidate which worth intervening. Okay, now since biology experimental science, we need to go for the omics data as well, and then got the omics experimental result for the what is the what we look into is in a virus we have virus related protein, virus associated protein. Okay, that's how the virus interact with the host side, right? And then we have like a molecule involved in the virus replication process, which we want to stop. Okay, so then well, we try to identify what's in between. Okay, you have like about like a 12 virus-associated molecule in influenza, and then a target biological process, which is the virus replication cycle. Okay, because we want to stop that. So what's in between? And that is a possible molecular target from the omics data aspect. Okay, that will be combined together with the control theory analysis of the molecular interaction map. So that's the intersection of that is a real target we should look into. So that's like in between, virus-associated protein and then the process from the omics data. And then control theory-wise, the structure network that actually meaningful to intervene. Okay, that was intervening. Okay, then what we need to do is then the target prioritizations. Okay, we have a possible target, and then we have a chemical library. Then we go to the docking simulation to be able to tell what target ligand pair is the was pursuing. So in order to do this, we come up with like a docking simulation. We bought like all the docking simulation available at time, and then benchmarked against the kinome assay. Okay, well, it's a kind of beta, right? And then we found out like a Pearson correlation of all that is very low actually. Pearson correlation 0.2425 and 3.3, and then Goes is the best is still like a 0.45, and the meta score 0.8. Okay, so it's not that great, but we noticed that each docking simulator actually had strengths and weakness. So we decided to cherry pick that and then assume if we can tell which docking simulator is good at predicting what for the combination, each combinations, what happened, and we found that actually it's pretty good. So we decided to create a machine learning system to be able to tell, given the target, given the chemical, which docking simulation we should believe, and what is the scaling factor we should use to do that. And then after that we oops, I just put this in animation so it's in detail, but like sorry about this, but like we got like a 0.85 which is a very high Pearson correlations. Like it was pretty good actually. And what happened is, this is another way of looking at it. So we verified in the kind of assay, and then a vertical blue bar is experimental result on the selectivity. Okay, selectivity is low if that is one is like it's interactive with everyone, right? And 0.1 is highly selective. So we just want to make sure selectivity, we get selectivity right. Okay. So the top dotted line is gold. So like gold actually computes like it's interactive with almost everything, right? A very low selectivity. And then experimental result is the vertical bar, and our system is like a blue line, which reasonably good estimate of the selectivity. It's very consistent with experimental result. Okay, and that's important because kinome assay and kinase pocket is very similar. So it's very difficult one as well. And then if you visualize this, and then for example, lapatinib, center is kind of assay panel. Lapatinib in our system is very selective to all the receptors. But the E hits gold and the autodoc and the Vena has the show the broad interaction outcome, which is not necessarily the case in the reality. Things like that. So like this is pretty good. At the same time, drawback to this machine learning thing is if you use the chemicals and target which is not in a training set, far from training set, performance is not good. But at the same time, like so like it's pity, but at the same time, it's in reality. You want to search through the chemical space in a specific category. So you don't really jump from the kinase inhibitor as a natural hub and something else, right? So if you do the drug discovery process and go to the kinase inhibitor, you search around the kinase inhibitor space, not something else. So you keep building data, keep training a network, and then within that space, you optimize that. You can have a very accurate docking simulation. That's a possibility. Okay. Now, after that, so we actually come up with the pathway, which the molecular interaction of the virus replication cycle, and then in-house database and a protein library and a chemical library. And what we did is the exhaustive computing on all the protein wherever the structure is available, not only the target one, because there's a network effect, the collateral effect. So against all the test compounds. So this is the heat map, okay. And then we computed the network effect. What is the most effective way of intervening in the pathway where the side effect can be minimum, okay. So we compute that, and we come up with a list of the top candidate, and then a few of them, and then we pick a 3 or 4 compound to go into preclinical trial as well, and then the preclinical trial going very well, and then we're going to mouse, and then this to monkey right now, and then hope to be able to go to clinical trial soon. Okay, now this can be applied into much more challenging one because it's getting more automated now to the herbal medicine. Okay, so Maoto, this is the herbal medicine, Chinese traditional medicine, and also Kampo in Japanese, and a mixture of the 4 herbal medicine, right? And it works in influenza. Okay, so we did experiment, all the assay on this. We got the the major pipeline of the identify the chemicals in extract and what's inside their plasma, and then what is eye-opening message is okay. I identify quite substantial chemicals from the extract and target, non-target the assay, and then what we discover in plasma is only fraction of that show up in the plasma, and then that changes over time. That's very, very striking, actually. So when you do that, either the food or herbal medicine, or probably even Western medicine, you really have to measure what's going on in the plasma, not the extract. Okay, and then this is one example. Is like a one with GL, which is one on the gray line, which the plasma peak concentration is in 10 hours after administration, right? The most one which are very quickly intake and peak in 1 hour or 30 minutes in 1 hour and going down. But a GR actually showing very different. The reason is it's going for the multiple step of conversion. So it takes time for the max out the peak in the plasma. And then also striking thing is, I'm sorry, that's the Japanese, but if you leave one herb out, and the efficacy goes down dramatically. So this is really the combinatorial. And the combinatorial in terms of 4 herbal ingredient has to be there, and each herb has thousands of active compounds. So we have to identify, and then it seems those are all important candidates. All the long tail size seems important. And we're getting all the clinical study and all that, because this is what we know already on the market. We're not going to the detail, but then we identified what's okay. So with that, that's a lot of work. We actually need to be automated. So we are now planning to build the fully automated biology lab in OIST, and then other place as well. And then we're working on the to do that. We had our data flow management using the Garuda, and I get the physical flow management, which we actually use the Garuda and then Unity software simulation package, and the material flow management, process flow management, that all combined into Garuda, and then we control the robot and control the human behavior, human experimental condition as well. And then I think it's very important to be able to automate this, because if you look at this, what's going on, in drug discovery, and this is something that we did years ago. Is this is a protein-protein interaction network. The B is on the right side is a human network, and then this is very consistent with the across the species. And then right side is K is a number of molecular interactions for each molecule, and then frequency is likelihood that that segment of the molecule is druggable. It became drug target. So a K20 and up is highly interacting in molecule, which is a P53, and it's very important. And because of the importance, the impact on the fundamental biological process, it's very difficult to be the drug target, because that has a lot of side effects. So all the drug actually which are targeting this is a cancer drug. Okay. And then the one in the middle, at the 5 to 20, is the sweet spot. Okay. But the interesting is that lower end, a K1, 2, it's rare, but it has like a certain probability that's go. So like a problem is like you have such a very large number of a molecule with this category, but like there are quite substantial drug target there, and because it's long tail effect, you can't assign a postdoc because likelihood that postdoc gonna successful is very low. But if you do that automatic, you can search to the entire tail end, and you have a lot of opportunity. It's a gold mine. Okay, in order to do that, I think we need the automation, the AI system, and then we're gonna create all the open platform system to be able to do that. And then this actually leads to the stage where we got the basic AI module, and then we now able to talk about the integrated module. Okay, we haven't got to the integrated module yet, but we have like all the then we talk like flow, data flow, and experimental flow, and protocol to be automatically generated and executed. So we're really working on the pass and then end-to-end experimental protocol design and how to manage that. Okay, and then all that. So I'm not going into detail. And then Garuda 2.x is a web-based, cloud-based system. So it's a multi-platform, large scale, and then you can actually go into multiple sites as well, and then all kind of management functions and knowledge integrations there. And at the end of the day, what you want to do is actually create a cycle of the getting from papers and database, generate hypotheses, and then design experiments, verify that. At the same time, what's going on is the accuracy of the report is the question. So like, if you got the knowledge extraction, how much you can trust that is actually questionable. So like you need, then still you have to reason that. So I would call this a right reasoning, because so you have to reason based on the very uncertainty place, which is correct or not, it's not clear. But during this cycle, that will be really the purified. That will have like an increased confidence on the outcome. Okay, now final couple minutes, and let me very quickly go through the further implication. And I think we can draw the lesson from the AlphaGo. Okay, as you know, the AlphaGo actually with DeepMind, as initial AlphaGo was using all the record of the Go play, and then thousands of millions. Okay, and using deep learning to be able to predict what's the possible next move. Okay, and then what they did is that they play against each other, AlphaGo against AlphaGo. So like with Markov random, a Monte Carlo tree search and the reinforcement running, so that they actually see like what a possible move, what is the second possible move, and then also randomize some move, and then go through all the search tree along the gameplay, and then able to evaluate which was a good move, which is not a good move. So like this, that's kind of reinforcement running part, right? And then what does this mean is that if you look at all the possible space of the Go, which is gigantic, and then past record of the human game is like a little red circle, which is only the fraction of that. Okay, so AlphaGo used as a seed and deep learning, use a reinforcement learning and Markov, Monte Carlo tree search to augment that and search probabilistic search of the possible vicinity of the game state and evaluate what move is good or not. So like by the time AlphaGo play against the best humans, AlphaGo already experienced a new outcome of the space which humans have never experienced, and then most likely that human move is within that area, and then so that's why AlphaGo is very stronger than human. That's no secret about that. Okay, what's interesting AlphaGo Zero is AlphaGo Zero starts from scratch, a tabula rasa system. So like no human record. So that's interesting. And then the AlphaGo very quickly gets strong, and it's a random play and the pruning, and then reinforcement learning, and of course combined with deep learning as well. And then I got the I think it was like first like 7 days, 7 days supersede the best human player quality, and then 2 weeks is better than the AlphaGo. Okay, it's interesting because why AlphaGo Zero get better than AlphaGo. Is that AlphaGo uses a lot of data. AlphaGo Zero didn't use the data. So I think AlphaGo is biased in a search space because they use the human data, which is like biased toward the human cognitive process. This is a kind of set of the play human likely to understand, human really prefer, or aesthetically or cognitively. But AlphaGo Zero, forget about this. AlphaGo didn't rely on that, and it purely search entire search space, or broad research space which is the best move for the game of Go, rather than what human perceive as a good one. So like that's interesting thing, right? Very different, and we can listen. That applies to the biomedical discovery. Is we need AI and machine learning for the we need modeling and simulation, because AlphaGo against AlphaGo is basically simulation, and then we need high precision experiments. Okay, in case of Go, that's easy, but like in that case, we have to low effort. So that need to be combined together, and then we can actually structurize the possible type of knowledge we want to discover first. Of course, there are other type as well, but like I think those can be discovered if you even if you stick to that. And then what the implication of those combined with AlphaGo lesson is we can start from the discovered knowledge, which is in the paper, and we can augment that. So that is actually vicinity of the what we discovered. That means human discoverable knowledge. I think we should start from there, because like that the way we can start. But at the same time, what's interesting is like if we keep going that, or then we might actually augment the space quite substantially. So that actually go into the territory that the human may not be able to understand, or maybe able to generate the kind of hypothesis. So like it's just interesting that if you machine are going to discover, we might actually see the discovery which we may not really understand very clearly, and we probably only are able to understand in approximation. Okay, so that's interesting. To do that, we need like all the data acquisition generation that be really going, quickly, and not just big data, but we need a virtual data generation. That's interesting. And in the case of Go, that was very efficient. But like in a biomedical science, any experiment science, how you do the virtual data generation and verify that's the meaningful one. That's a challenge. Evaluation is really the challenge, but that's actually the key. Okay, okay. Then I just then you might ask a question in science. Asking the right question is very important. Now, asking the right question is very important because career path, time that you active scientist is limited, 30 years or 40 years maybe. Okay, but if we are to be successful, you have to ask the right question so that you can actually be successful within the lifespan, within the duration of your active scientist, and in the resource that you can get. But if you automate that, okay, this limitation is not going to fade away, but you can ask every question, a lot of questions, and then some of the important questions and important answer may be in that. So like asking the right questions, although still we have a resource limitation even in the machine, not going to fade away, but we have a very different power of asking every questions, and we'll ask many questions, important questions in there. So we may be able to ask a question which we haven't thought about it. So you go back to the redefine scientist guy. This is go to the massive search and other verification. And the question is, do we search entire space or search the fragment? So if we have to have fragment, for the specific areas, and we let the characteristic AI and specialized AI to communicate to each other, or one universal AI. That's kind of interesting questions, which is more efficient. But like if we consider population characteristic AI scientists will be their way to go. And then each AI scientist going to have a characteristic. They're going to have a specific order of discovery, which may mimic the very successful scientists. We'll see if there's a natural order scientific discovery, which probably it be, and then whether this will follow that. We look at looks like a career path of the successful scientist, and then we'll start understanding what's serendipity by accident. So that would be the important question. So implication, the final slide is if we do this. This will result in alternative form of scientific discovery, obviously, and probably alternative form of the intelligence. And we will be able to accelerate science at unprecedented speed, which is very important. And an interesting question is, do machine evolve by itself? Perhaps yes, but that will actually pose a great challenge or a great opportunity for our civilization in the future. That's it. Thank you very much. And I'm happy to take questions. Okay. Okay, so we have questions in a chat window, actually. Given
yes,
that
we have questions, yes.
Yeah. Do we do interactivity, or should I just answer from all the chat?
Yes, if you can, please read the question before answering it.
Okay. So let me start with the first one. Given that the AI search engine can explore the hypothesis space defined by the mental model, please comment on what you envision will be the progression of computation tools in the workflow starting the data science question. How can such tool be put into the hands of the many data scientists who only have access to limited computer resources? Well, I think like I don't know. I mean, let me understand the question properly. I guess like so the okay, so availability tools, right? But basically, okay, I think it's quite a bit of limitation at this point, but I think like we'll start having the more open data sharing once people understand this will push the science, and obviously people wants to move the science forward much faster than today, and I think given the tool like this, I think we want to have like a more organized data collections and then data publication sharing scheme, as well as the software curations and data curation as well. And then there's a lot of discussion going on some of the publishers on will actually do that. So I think data resource will be available in the future, although it's not really entirely, but like I think it's going to be much better. And the computing resource, well, that's still the bottleneck, but I think I think there will be some sort of public resources for that once we get there. Like because people's not aware of this is possible. I mean this is on the way. Like 5-10 years, I think it became obvious. This is gonna be really critical for data science. Okay, the second question. Can you please describe computing environment requirement today for this research and what is anticipated in the future? Okay, do you feel that the supercomputer is required? Yeah. We use a supercomputer. Obviously, we use a supercomputer and we use the AWS, and now we use like GPGP cluster. And particularly, we use the supercomputer in the University of Tokyo, and then we start using the GPGP cluster, ABCI, and then AIST. And then have like the local cluster as well. So if we can actually tap into the supercomputing resource in your side, and we can work together, have like a gigantic supercomputing resource, and then data resource, combined together with our algorithm and then setup. I think this is really good. I think it's computing is key. Data and computing and proper algorithm is the key. Okay, for logic analysis, which system are you using for the logic reasoning? Open. Yeah, PLN NAS. Okay, Garuda use multiple tools. Okay, so it really depends on the setup or like users it will Garudify that and use multiple platforms. So like point of the Garuda is open. So it didn't stick to that. I use this tool or this analysis, but like a variety of analysis tools and then a method should be on board. And then that will be selected by use. But Garuda actually has a recommendation engine which algorithm to use. This tool will be proper for the specific data type, and that can continue. Okay, and then what about IBM Watson? Okay, yeah. You know what's going on, right? And then Watson's a great idea. At the same time, in order for the Watson to be functional, we have to do a lot of data curation and then the building up. I mean, Watson is not really the one integrated software. It was a library of the AI algorithm and the modules, right? Yeah, so you have to do a lot of effort. I mean, okay, sometimes discovery outside the target field research can have a significant impact. Yes. How could this? Yeah. So like that's why I think we need a different character of AI system in a different area and let them talk to each other. I think that one way. I mean, rather than might have like everything together, that could be possible. But I think that I think the search will be very ineffective in that time. So like if we have like multiple characteristics in the AI system, so the society of AI scientists and talking to each other. Of course, like a human can get involved as well. I think that. But at the same time, go back to the initial claim. This system, at least in the early stage, not going to discover everything, right? So we discover, what we're saying is that, with a specific kind of discovery, which is good at, this system can be good at, there will be the major discovery coming, okay? It's not really claiming every discovery will be done this method, because we're not exploring all the discovery space. But at the same time, search and optimization space and specific structure of discovery, we can discover a lot of interesting thing because of that tail effect, and then also all the human cognitive limitations. So I think from there we will have a sufficient push sufficient paradigm change in the science, even though this system might not be able to discover everything initially. And then if we're done, we can actually explore the other type of discovery and then expand the capability. Okay, and then of course that CRISPR-Cas was one example for this question, which is very tough actually at this moment. We have to think very carefully how to do that. Regarding recent study published in IGP transaction, which refers to medical diagnosis, did you compare your test solutions with one of the? No, I don't have access to IBM Watson, so we have to be able to access Watson to be able to the benchmarking it. But our system is still making a lot of progress at this moment. I hope to be able to do something. But IBM Watson is basically is actually data access and inference from that. So they have the best one is cancer therapy choice, cancer genome, and that it's great because they work very closely with Sloan Kettering, right? So they have all the literature and a database of the specific type of subtype cancer and what is the possible therapy options. So it's basically a very intelligent database search and a pattern matching. So Garuda is open source. It's not necessarily open source, but open accessible. And then with the specific partner, we can talk about how we actually disclose the source. One possibility that we might actually go to open source in the near future, but we haven't made a decision on that. But for the academic establishment, we can actually distribute the Garuda baseline for the free, and then we can work together on that. Okay, so do you think that the narrow AI just explaining experimental data will suffice to make a discovery worthy of Nobel AI, or be necessary to build a fully general AI to achieve the purpose? Do you think a general AI will be created in 2050? Okay, I think broader competence, but not such general AI will be able to discover. I think human, it's not a general competence, but if you want to be, expert, you have to specialize it basically, because of the resource limitation, amount of time you have to use for the training. So I think for AI scientists, I think it will be a scientist. So they may have some knowledge on the other field, but probably predominantly science. But of course, if you want to have some unexpected discovery, you might have a much broader knowledge. So probably it would be great to have more general AI, but I think a specialized AI will be able to make major discoveries. And then making the scientific discovery probably doesn't require a general AI. Of course, that really depends on the type of discovery. But again, it's come to the point I'm not really claiming to make every discovery to be made by this system. So in that case, you can really focus on the what machine could be good at the specific time of the stage of the technology. Okay, 2050 general AI, I don't know. I have no clue because 30 years, I hope we can have something, but at the same time, depends on general AI means a mimic human being, then I think it would be probably cost effective to hire a human. So there's an issue of the investment on the research in that directions. Okay, do you try to use the AI docking system to predict the optimal binding site for the valence docking from the chemical bond and the target? An example, the bottom is no, I don't think we have done that. We actually focus on pretty much on the kinase inhibitor and then herbal medicines. Herbal medicine system based on the training data and the kinase inhibitor was not very good. And then we have started looking into the herbal medicine natural compound database to be able to retrain that or augment the training data set to be able to do that. Okay. How can AI and ML be used to answer the key fundamental question in the biological science? Those specific limitations too? Well, that's the whole point of this talk, and we're trying to understand that question. I hope we'll be able to, fundamental question in biomedical science with the approach we have. A lot of fundamental, it really depends on the fundamental biological question, but a lot of biological questions, which sounds important, it's about the process and the mechanism behind it and the who's actually involved, which molecule to be involved. So if we can actually solve that question in a large scale, I think we'll be able to have a lot of interesting critical discoveries, actually. And then beyond that, that would be interesting question. What's beyond that? I mean, then we might actually have to have some sort of style of discovery as well. But I think like we can expect a lot, but I think we can a practical starting point would be to have a specific type of discovery, but a large scale, so that where the human cannot really do sociological, resource, and psychological reason, and then discover something, because like you never know what's important, and then if you go to the relatively seemingly unimportant important genes and molecule, less likely that you want to pick it up, but it could be the important one. Again, how you do that is the most best way to do that is the machine to be able to make a discovery related to that kind of molecule in a large scale. I think that's really the one of the strong points of the machine. The human won't be able to do that. Okay, I think that's it. Or is there any more questions? I think that's what I see. That's all I see in my screen. But okay. Are we done here?
Director Marek, can you hear us?
His screen seems to be freezed.
I will reassign. I will reassign his presenter role.
Yeah.
Dr. Kitano-san, thank you so much for wonderful talk. I do have a few questions, but maybe I'll ask you. So, do you believe that by 2050 we will reach some sort of singularity in the sense of Kurzweil, that we sort of shoot off and reach completely unprecedented heights? And the speed of discovery?
I think, yeah, in terms of scientific discovery, I think we will reach the some sort of singularity. I mean, doesn't really means like AI system to be better than every type of discovery, but I think AI will be indispensable part of the scientific process, and then probably we will rely a lot on the AI systems. What the AI system discovered for us. I guess it would be, I think like any top-level research institution without AI scientists would not be competitive. I think that will happen like way before 2050.
Excellent. Just to comment to some of one of the questions, one of my big hopes in our interaction and projects with the Systems Biology Institute is that some of the scientists in Poland or Warsaw perhaps will have access to Garuda and Textila, and will be working on extension of some tools, especially Textila and in machine learning space. So if that would be very exciting for us. Thank you again. That was a wonderful talk. We are so happy to have you. Really very grateful. And thank you. And now let me just remind all our listeners. First of all, thank you for being here with us, being present at our seminars. I see lots of names of many friends, and almost every person who asked the question is a friend. So I'm saying hi to all of you. If you happen to know some really fascinating visionary thinkers who work in the field of computer science, please let me know if we might consider inviting them. I don't have a firm speaker for next month. Maybe we'll skip. I'm not sure yet, but we'll give at least 1 week or maybe 10 days advance notice. It might happen on 16th of December, maybe later. If not, then I welcome you all in January. And again, I've acknowledged support and partnership with various media outlets. They help us to reach very large audience. I encourage you to check our website, where you can, at your leisure, listened to all the previous lectures, and I can assure you that they were equally challenging, exciting intellectually. So I'm very happy that we have such an incredible set of speakers, a group of speakers here at the seminar. So we'll try to keep that level. And with that, thank you very much, and let's see each other next time. Thank you. Bye-bye.